Hi, welcome back to Physionic. Today's topic is going to be, unique from many of my other topics that I've ever discussed, although it seems like it's a regular topic, discussing how saturated fat may impact the health of our cells. So yes, I'm going to be going into some molecular biology and some cell biology and investigating a particular study. So that is all normal, as you may know if you've been with Physionic over the last 8 years, but something is unique about this study in that I think it may be one of the worst studies that I have ever read in my life. And then you might be asking yourself, well, then why would I cover it? Well, I think it's important, specifically because one of the major areas of this brand of Physionic is to educate people on how to read studies and understanding that we can't just, this is a prime example of why we can't just look at the abstract of a study and then just assume that everything is hunky-dory, which is what a lot of people do. A lot of the gurus out there, they will read the abstract of a study and not critically analyze it. And as I've been weekly for many years now, analyzing studies and breaking them down, and I've had my small qualms with certain studies. I've had maybe larger qualms with some other studies. This one, I have some absolutely epic qualms. So, that being the case, I thought I'd go ahead and present to you some of the issues and some things that you should look for when you're dissecting research. And if you're not familiar with who I am, why you think that this pipsqueak might have any level of authority to speak on this topic, my name is Nicholas Verhoeven. I'm a doctoral candidate, PhD candidate in molecular medicine, so I have a background in all this. My concentration, if you want to call it a concentration, not that physiology really is a concentration, considering it's applicable to all areas of medical science, is cell physiology, molecular and cell physiology and pharmacology. So with that out of the way, let's go ahead and jump into this study, and hopefully you'll forgive me. Maybe we'll learn something. Who knows? Maybe we'll learn a thing or two, but it's more so about the analysis of the study as I go through it with you. All right. So I don't even have notes for this because I can just do this off the cuff, just because it's just really bad. There's so many things to say, but it's not too terribly long, so don't expect this to be massively long either. So the question that we're going to be examining is how does saturated fat impact your cell's health? And there are going to be a number of different metrics to determine cellular health. Okay. So this comes from a paper called Multiple Pathways Are Involved in Palmitic Acid, which is palmitate, which is a saturated fat, the most common saturated fat, induced toxicity. I don't necessarily want this to be a reflection of these researchers. It's just more so of this general idea that just because it's a study means that it's necessarily good quality research. And this comes from a very low-tier journal. And as a matter of fact, I'll quick throw this out there. I am a big fan. Let me quick go full screen for this. I'm a big fan of the Joe Rogan podcast. I know this is completely out of the left field right now, but I'm a big fan of Joe Rogan. He's a fantastic interviewer. And while I don't agree with everything that he says or everything that he espouses, he had a particular guest on that people have been showing, just kind of sharing a lot of his content. And his name is Peter McCullough, something like that, something along those lines. Anyway, I listened to about 80% of that podcast where they discuss the virus, the pandemic, et cetera, and they discuss other things as well. And this particular researcher, I promise this is related to what we're going to be talking about. This particular researcher likes to have it known that he is the most published researcher in his field. And I decided to do a little bit of digging. And I may actually make a particular video on this because this is an important principle for people to learn. And it turns out that this researcher has published in some of the shittiest journals I have ever heard. As a matter of fact, if anybody wanted to do a little more digging, they would find that he is actually the editor-in-chief in many of the journals that he has published in. Do you think that is a conflict of interest? Pregnant pause right here, because that is a massive, massive conflict of interest. But hey, he's a reliable source. I don't know much about him other than that. I just looked up that one interesting facet because he seemed to be bragging about it quite a bit, that he is the most published researcher in his field. Well, it matters on the quality of the journal as well that you end up publishing in. All right, back to it. So this is in a really low-tier journal, extremely low-tier journal, but still, just because it's in a low-tier journal doesn't necessarily mean that the data is going to be crap. However, in this situation, it does turn out to be the case. But again, this isn't a reflection on necessarily the researchers themselves. I'm not speaking to them. I'm speaking more so to the general population and understanding that we need to not just read the title, not just read the abstract, but actually critically analyze the data itself. All right. So the first piece of data, this is what they did is they added. Let me get rid of my. Okay. What they did is that they had cells, CHO cells specifically. It's not going to matter too much what type of cells, but they have cells. And then what they do is they add increasing concentrations of palmitic acid or palmitate saturated fat. So this is in micromolar amounts. So they add 31.25 micromoles of palmitate, then 62.5, 125, and 250. And our control condition here has none that has been added. Then they measure the amount of lipid droplets that occur. And I created a diagram here so that you can see what lipid droplets look like. They're essentially just, you have these fat molecules and they can be sometimes stored in these droplets of fat. And it is not accurate to say that lipid droplets, these droplets of fat, these sacs of fat that are found in our cells, are necessarily negative. I have content where lipid droplets actually turn out to be rather beneficial. But in general, if you're not talking about like an exercise condition, typically this is not a great sign. So palmitate, how much of it ends up getting stored as lipid droplets. So you can see the control is very low. Then as you keep increasing the concentrations, although here we don't find significance, but here at a decent amount of palmitate that's been added at 125 and 250, you see that there's an increase, a kind of a stepwise increase in palmitate being stored, or at least the formation of lipid droplets. So that's interesting. You know, I've actually had this corroborated in multiple other studies that I've looked at, but that's not the point of this. I'm just looking at this real study, but I may mention that from time to time. This isn't overly alarming. I do think that ultimately, if I can zoom in here real quick, I do think that they probably should have done a slightly better job with these asterisks, just because you're putting it right on top of the bar, but I am totally nitpicking there. So I'll leave it at that. This is relatively okay, not my way of explaining things. Now, the next thing that they wanted to look at was ATP production. So more specifically, probably looking as a measure of mitochondria. And so not that ATP is just generated by mitochondria, but it's by the majority produced by the mitochondria. So what they've done here is, again, we've got our control condition, and then they've got ATP production, or the percent of control. So these are compared to the control condition, like that. And here we've got no saturated fat added, and then we've got, again, the same stepwise increase in saturated fat. That's fine. But here's something that's rather odd, which is completely unnecessary. It's a little bugaboo. It's not a huge deal, really, but it's kind of a comical thing to throw in here. Typically you have, so if you look at the axis here, you have 0 to 10. This is all percent. So 0, 10%, 20%, and then they've got this break, and then it picks up again at 80, 90, 100, 110. Okay, that's fine. This is normal to do that in research, but the only times you would want to do that is if you have values that are in this 10 to 20% range. There's no point to have this. You might as well just change. You might as well just do like 100. It just doesn't add anything to this data set to have this break in here. It's just kind of pointless. So is it a big deal? Maybe they wanted to add, I don't know, they wanted to have this granularity between 100 and 90, but then in that case, you can just have 100 to 80 and then just, that's it. And then just show the difference between there, because then the assumption is there's nothing else, which there is nothing else below there's nothing even below 90. So is it a huge deal? No, it's not. It's really not. But we're going to get to the bigger deals. Don't worry. This isn't me just crapping on a paper for just to crap on a paper. Okay, the next thing here. Okay, this is where we're starting to get in kind of the bigger deals here. So here what they're doing is these are microscopy images. So this is fluorescent microscopy. This is similar stuff that we do in the lab. And here they're looking at 3 different proteins. Really, they didn't describe them as 3 different proteins, but I'll get to that in a second. So here on the left side, these 3 images, 1, 2, 3, are all without palmitate added to the cell. So this is in a control condition. Now, these 3 on the right are all with palmitate added to the cells, and then they see what happens, what's the change, right? So here they're fluorescing. So the blue is most likely what's known as a DAPI stain, which is I guess I should get rid of my, is called a DAPI stain. And typically that's for the nucleus, to stain the nucleus so then you can see the, what's inside the nucleus, what's outside the nucleus. And here, this protein called LAMP2 is related to autophagy. It's part of the phagosome or the lysosome. And it's just essentially part of autophagy. That's really the big point that I'm trying to get across here. So the point is that you compare this image to this image in terms of the green. Now, unfortunately, they didn't actually label this in the paper itself, so I'm just assuming here. But the assumption is that the green is LAMP2. And the idea being that if you have higher levels, then you would have more of it all over the place, or it would be brighter in some way. So if you just compare those 2 images, sure, I would make the argument that there's more LAMP2 with the palmitate addition. However, the big bugaboo here, the big issue here that you can't have unless you have really drastic differences, and even then it's typically not recommended to do this, but unless you have really drastic differences, you should have a quantification of this, because it's easy for a person to just say, oh, well, I want to believe something, so they just see this and they're like, okay, well, I don't like saturated fat, so I'm just going to assume that LAMP2 is highly elevated, meaning that there's more autophagy. Now, granted, people think of autophagy as a positive thing all the time. Well, autophagy can also cause cell death. Bum, bum, bum. Yeah, that's right. Autophagy can actually cause cell death as well. So having overexpression of autophagy can lead to this cell death. So the idea here is I'm assuming that LAMP2 is elevated. But really, if you squint your eyes, maybe you might not tell the difference. Now, for calnexin, this is another protein, so they're expressing it in green, presumably again. And you've got your DAPI stain. So with calnexin, this is a protein that's found in the endoplasmic reticulum, which is another section of the cell. And it is typically aggregates and tends to be increased when you have what's known as endoplasmic reticulum stress. So when you have stress or the buildup of proteins within, or fats or whatever it might be, within the endoplasmic reticulum, then you get this increase in calnexin. So again, if we compare between the 2, sure, there seems to be an increase in calnexin, but it's not that extremely obvious. Here it seems more diffuse. You know what? Let me go ahead and zoom in here a little bit more. Yeah, so here you can see it's a little more diffuse, but you're also looking at 4 different cells. Here you're looking at 2 cells, but calnexin seems to be more expressed. But again, you need a quantification here. This isn't clear enough to be able to determine immediately that calnexin is highly upregulated with the palmitate condition. And finally, this is what I was going to say something in, but so let me back this up. Okay. This is a measure of mitochondria. Now, in the paper, they actually say this is a measure of mitochondria. You don't say that. Well, you can say that, but in addition to that, you need to say what's the mitochondrial marker that you're using to test or to measure mitochondria. So is it something like TOM20, TOM44, TIM proteins? Is it MCL? Like, there's a bunch of different OPA proteins, MFN. There's a lot of different proteins that you could use to measure cytochrome C, to measure. I think maybe eventually at some point they say cytochrome C, but in the legend they end up saying it's just a measure of mitochondria. Okay, well, again, here, when we compare the two, it's really difficult to tell. Like, is there a decrease here? Maybe there's a decrease here. I think that's what they're trying to get across, that there's a decrease in the red, which is the mitochondria. Again, assumed. But you need a quantification to actually be able to tell if there is a difference between those two. And the quantification needs to be probably per cell as well. So because you've got so many cells here, and then here you've got far fewer cells. It's these kinds of things that you have to look out for. Otherwise, it's really difficult to establish the differences between them. So I'm still sort of nitpicking, but this is kind of a big deal. You need to look out for this. Now, this is where things go really off the rails. I'll give you an example in the next slide, but here again, we've got our same conditions. We've got a control condition without palmitate addition. Then we've got palmitate at these different concentrations as it's increasing. Here we've got a measure of the actual protein amount. This is what's called a Western blot analysis or electrophoresis. And here we're looking at the exact calnexin amount of the protein that's found inside the cells. That's fine. Okay, so control. Then you've got maybe a little bit more with the addition of palmitate. Then it seems like it kind of decreases. Then it sort of maybe is the same, and then there's a slight increase. And the way I'm measuring that is simply by looking at the darkness, and secondly, the kind of width of the band. Now, that's fine. The results are the results. So even if there is a decrease here, that's just the way it is. And it's honest of them to present that as it is. Then they've got another measure for LAMP2. And if you're thinking that this looks really messy, that's because it is. This is a very messy blot. And another thing that I'll have you pay attention to is that the levels don't change here, the levels don't seem to change here, the levels don't seem to change here, and then suddenly they increase substantially for these two conditions. Now, the problem here is that one of the problems is that the background is really gray, so it's starting to get to a darker gray substance. And that means that they really had to expose this for a really long time. So they had to, if you end up doing Western blots, you'll understand this much better, but meaning that they have to measure this multiple times over. So increase kind of the magnification of the signal multiple times over just to get this measure. It's not a huge problem, but the problem is that these two are measured variables, meaning that they are things that are going to change with the change in these conditions potentially. What they need is a control variable, meaning a protein that will not change. So this is called a loading control or a housekeeping gene. So you would have something like actin, you would have something like tubulin, you would have something like GAPDH. You have a series of different proteins that they stay pretty stable no matter what the condition is. So with each one of these, you have a change in the condition, right? So there's not much change here, but then you have this massive change once you get to here. Or you don't have much change here, but then you have a pretty big change when it comes to 250 micromolar of palmitate addition. So you need another condition here that does not change based off of the concentration of palmitate that's added. And the reason for that is because you want to know, you can have a situation where a person, this is really shady, and people shouldn't ever do this, but it has happened in science and they usually get caught. But you can have a situation where a person will load a much smaller amount for something like the control, which would show up if they had a loading control, but it may not show up here. So that's how they get a lower control. And then they can load a lot more of the total protein, the total amount of cells that they end up loading into this experiment for these 4 conditions. That will then increase the expression here, and then you would have a comparison that is unfair or inaccurate, in that the loading was not the same. You need to have the same amount of total protein to measure within that total protein how much LAMP2 there is and how much calnexin there is. Now, I get that this is kind of a nebulous idea, and it's difficult to explain. So I have an image to show you this. All right. So this is not related. This is not part of the study. This is actually something that I just found off of the good old Googles. And it doesn't matter what these proteins are, but I did mention that one of the loading controls would be something like GAPDH. GAPDH is not a protein that changes very readily. So as a result, you can use this protein to be your loading control. So in this situation, you can tell that the differences between these four loads, or these 4 conditions, it's pretty similar, especially these 2, but even these 2 are pretty similar to one another. And you would still quantify this, like you would still find the density and you would still try to quantify so you actually have numbers. You don't just base it off looks. But if you were to just take a quick look at this, you'd be able to tell, oh, okay, this is loaded equally to this one, and this is loaded equally to these 2 as well. And you see a decrease in, I believe this is cadherin, but I'm not sure about the protein. So let's just assume it's cadherin, the protein. So you can see that there's a decrease here and there's an increase here. So it's not because that they've overloaded this one or that they've underloaded this one. They've loaded them all the same, and there just turns out to be an increase in cadherin and a decrease in cadherin. Here, no loading control. There should be a loading control under here so that I can tell if everything has been equally loaded. That is vital. This is a huge no. This immediately, the data just goes out the window because you are not showing the loading. You have to at least show the loading, and then what's even more preferable on top of that is to show the bar graphs to actually show that you did the numbers and then compared the numbers and normalized the numbers, et cetera, as you're supposed to for statistical measures. But just simply looking at this, you can already tell that this should have had a loading control. Big no. All right, so another thing here is they looked at cell viability, and this is a measure essentially of how well do the cells survive over time, over a set amount of time, like 24 hours, for example. So again, we've got our control condition. All of this is percent of control, which is well done. This is exactly how you should do it, because you've got your control condition, and then you're going to compare all of your manipulated conditions to that control condition. So this is well done. This is good. And here they're showing that this increase in palmitate or saturated fat, and you can see again it's a dose response. So you're seeing a gradual decrease in the number of cells that end up surviving. So we've got a healthy cell, and then it's turning into what's known as an apoptotic cell, so it causes cell death. So this is fine. Okay, so this looks really complex. It's really not. This is, I'll go ahead and explain that this axis right here shows, so the y-axis is showing just the number of cells. number of cells. Doesn't matter about anything else, just know number of cells. And then here they're showing the stages at which the cells are found in their mitosis. Mitosis is cell division. So when the cells are dividing, when they're exposed to these different conditions of palmitate, increasing levels. Again, this is our control condition, so no palmitate, then increasing levels of palmitate addition. What are they finding? Are the cells being stuck in a particular position, in a particular phase of the cell division? So one of them would be sub G1, which is kind of a sub phase. It's not really very commonly talked about. It's usually G1, then the S phase, and then G2 or M phase or mitosis phase. So G1 would be a growth phase. So the cell is producing more proteins in preparation for it to split into two cells. Okay, so let me, I know I'm covering up one of the, so in G1, you've got the production of a bunch of proteins, and the cell is still one cell at this point. Then in S phase, here, this synthesis phase where you get the production of DNA, so you get the replication, because if you're going to make a whole new cell, you have to make copies of every single gene within that cell. That's the S phase. So those cells are replicating all of their DNA, making copies of everything. And then at that point, then you have doubles of everything, so you can split the cell apart into two cells, and then you have one copy of everything, well, sort of one copy, just based on our discussion, of everything for each cell. All right. And then which is the M phase, where you have growth again for a while, and then you have the M phase where you have the actual splitting that eventually happens. So and really all we're doing here is comparing these graphs. That's all you need to really know, and that's it. And here they've done the quantification, presumably for so how many cells are found in G1 versus S phase versus G2 M phase. All right, so when we look at control, you don't need to focus you can unfocus your eyes and just look at, are there differences between these peaks? Is the peak height different? Have things shifted over? Is it more in S phase compared to G1? Things like that. So control with this low palmitate, kind of middle palmitate, higher palmitate, it doesn't seem like there's much change. Maybe there's a slight increase in the S phase and a slight decrease in the G phase right here. But these three are relatively the same, so it's not showing any sort of critical differences. But then once you get to the really high palmitate addition, then you can even see it here by the quantification. You have more cells, so if you compare this to this, you have more cells that are found in the G2M phase, and you also have fewer cells that are found in the G1 phase. So what does this tell us? Well, I actually had to look this up because I wasn't that familiar with how to read this in terms of what does this mean? And it turns out that if you have more of these cells that are getting stuck here, or you have fewer cells that are in the sub G1, or maybe it was more, I can't remember. But if you have more of this characteristic, then you're getting more cells that are going towards apoptosis or getting more towards cell death. Is this a huge deal? If you just crank up the amount of palmitate that you're adding, I don't think this data is just that great. Let me correct that. Let me say this is good to see that they're showing that, okay, even though there aren't any dramatic differences, we're still showing you the data. That is excellent. Great. But there's no real differences. Do I think that this is something that's really damning evidence that saturated fat leads to really destructive things inside the cells? No, I don't, because this is such a high amount of saturated fat, for example, that I'm not really sure if it's because of the saturated fat at this point that we're getting this effect, or if you're just overwhelming it with just molecules in general that's leading to this effect. Now, if we saw stepwise change like we did earlier, then I'd be certainly more convinced. And we did see that in other data, but this data just doesn't convince me. Okay, this is the second to last that I wanted to cover. Here, they're looking at the reactive oxygen species generation. This is oxidative stress. And here, again, we see this stepwise increase. And this is a perfect example of what I was talking about in the very first piece of data. So here, we've got a control. This is our, so yes, we've got our control. Then we've got the addition of all the palmitates, so the saturated fats, to the cells. And we see that there's a stepwise increase in the generation of reactive oxygen species, which if you have too much reactive oxygen species, as I have diagrammed here, then you get cell damage. And obviously that is not great for the health of the cells. So there's a stepwise increase. And then they, just as a what's known as a positive control, they added H2O2, which is a reactive oxygen species itself, or a free radical. And this, of course, if you're actually adding reactive oxygen species to the cells, then of course there's going to be tons of damage. I find it funny that the image Ross, reactive oxygen species leads to Nick. Anyway, back to this though. So if they're adding a ton of reactive oxygen species to the cells, they're just showing that, hey, our model works, that when we're measuring ROS, that it works. So we're not just measuring random nothing. And of course here, right, the amount, this is percent of control. So the control is right here stuck at 100% obviously. And then we've got the stepwise increase. However, and this is why they've got this break here. So they've got a break here because the addition of just pure reactive oxygen species is so much ROS that it's going to be off the charts almost. If they had stuck to this gradual increase of 100 to 200 to 300, this would have been just through the roof. It would have been flowing through the graph. So they had this break here that they introduced, and they started it back up at 500, 600. This is an example of when you would want to use this kind of break. The first one just didn't make a whole lot of sense. Okay. Now the final piece of data is, I don't like this at all. Because, and I've got this shown. They added the arrows, and I added the circles because I needed things to be more clear. And then I added this because this is what the circles are pointing to. So this is what's known as electron or light micro. This is light microscopy, but it's electron microscopy. And here, they're just pointing out what are known as phagosomes, which is what I was sort of talking about earlier with LAMP 2. So phagosomes are kind of the precursors to autophagy, and they're these sacs that will essentially envelop components within the cell. And once they do that, then if a lysosome binds to them, then they end up destroying the things that are inside. So what they are pointing at, supposedly, is phagosomes with lipid droplets. The problem here is that I'm just taking their word for it. I don't, like, if I'm not, even if I'm an expert in this, this doesn't necessarily, like, I don't really know what I'm looking at. And I've been looking at this stuff for, a while now. But even so, the problem is here, this whole image is just the palmitate condition. So they've added palmitate just like they did in their other experiments, and then they're just taking images of what the cells look like. So this is what cells look like. This isn't, cells aren't always like round and just, very nicely shaped. They look kind of gruesome sometimes. That's not anything that they've done wrong or anything like that. But if you zoom in here, then they have these arrows that point out, and I'm gonna zoom in here. So let me get rid of my face. Okay, so here we're zooming in, and here are these phagosomes, and somewhere inside of them there are these lipid droplets. However, what would be far better to do would be to label the lipid droplets, and I don't mean them as in like just have arrows pointing them. Oh, I see that I've got one mislabeled here. So this one is actually supposed to be over here. But it's not just labeling them in terms of like, I'm going to have arrows pointed, like actually use things like immunostaining. So you would use antibodies or something along those lines, or find some sort of staining that can actually give you a representative image, so you can have colors that show the different sections of the cell. This gives the reader, the person analyzing the data after you, an indication of, oh, yes, okay, I see what you're pointing at. This actually is what you're saying it represents, because the label only works on that thing. So I think in previous papers that I've covered, they've done something like immunogold labeling, and you can use other types of labeling as well, but it can work in this kind of context. But my bigger problem here is that they're showing us this picture, which is a cool picture in its own right, I guess, but they're showing us this picture, but they're not giving us a comparison image. So here we just have the palmitate condition, but we don't have a control condition to then compare the control condition to this palmitate image. That's a big deal, because at this point right now, this is just kind of cool picture. That's it. Like as a person analyzing this, you would just say, cool picture, bro. That's it. Okay, so my conclusion from this is palmitate does bad things to the cells, supposedly. Based off this data, we can't really say a whole lot. Some of it, sure, but I'll be covering a lot, I think, better papers than this in the future, in the near future. So, and the data is so bad that I just essentially trust none of it. So this is a great example, folks. This is why you need to read the papers that you're citing. When you follow people, certain individuals that I will not name, and they just throw up the abstract, it should give you zero confidence that that they've done things well, that they've done they've done you a service, that they've actually done a reputable job of analyzing the data. And even when you analyze data, you can get things, you can miss things, you can get things wrong. And that's the point. That's the point of this. Physionic. Not me, Physionic. I am getting better and better at analyzing data from papers, and I deeply, deeply enjoy it. It makes me so happy to do it, and I have a ton of fun doing it. And that's why we have these long-form videos, so that we can do it together. But as you become better, as you follow along, that you will pick up on things, some things that I will not pick up on. And that's why when you comment and you mention things and you push back, or you point out things that I missed or that you disagree with, that allows me to make amendments to, and I always have an amendment section, and I always pin the comment to make sure that people are aware, hey, there are things that I've missed, or there other feedback that other people have mentioned that you should know about as well. And if it's really bad, then I take down the video and I redo it and I'll post it back up. That's like trying to be intellectually honest about how to read literature, and I would say that a vast majority of the gurus out there that talk a big game don't have the slightest clue how to read this kind of stuff. And that's why they just go off of the abstract. So that whole soliloquy out of the way, I will leave it here. And thanks for stopping by. And next week, we will be back to reading a little more substantive papers, although the papers that I've selected, I have a few issues with them, but overall, they're pretty good. So we're still going to get some really, really cool information actually related to this topic, saturated fat and its effect on muscle health, mitochondrial health, insulin sensitivity, muscle health. It's going to be really interesting, so stay tuned for that. All right, with that, have a wonderful day, and I'll catch you in the next one. See ya.